Well, you promised a lot now on my behalf. I'm not sure I'm going to fulfill all those expectations now, but something. And thank you very much, and thanks for funding it. That's Katka who actually brought home this little grant what we used to bring in our guests now and then. Also going ourselves to Oregon now and then. There will be a great meeting of 2D spectroscopy this summer there, and the whole or half or how much, quite a lot of us is going there. So you will see us soon again after you go home. But Katka was pretty much right when saying that I got interested in coherences long time ago already, and this interest has evolved towards various directions, and one of the directions is how do those coherences live in very single systems, for example. Is it the same experiments what you carry out in a single system versus when you do in a bulk? And what you can see here is a typical photon echo type of signal from a bulk. And the point is that what you see is a phase-matched signal in different directions. Here are the laser beams coming through. Here is, for example, 2-photon echo or 2-pulse echo from these 2 pulses. Here is a 3-pulse echo from here. There are various high order signals still, but the main important message from the figure is that in order to get phase matching, you have to do the bulk signal or bulk experiment. If you want to do that type of photon echo type of measurement, you can't do it in a small system. The system has to be bigger than your wavelength. LH2 is much smaller than that. LH2 light harvesting complex, for example. So it all started, well, it started before, but the big fuss started when the oscillatory signals in this so-called 2D spectroscopy, photon echo-based 2D spectroscopy, were observed. The method was developed slightly earlier, but then soon it was seen that these things, those 2-dimensional features, they wobble up and down and become broader and narrower. So depending on what you look, you see various oscillatory signals. And it was then assigned to electronic coherences. And some speculative sentences were written at the end of this article, which attracted attention for really many people in the coherence field. Quantum coherence became a very important topic. Lots of headlines in prestigious articles, even in popular literature. It was the parallel quantum pathways were discussed in a novel where there was a discussion between a Nobel Prize winner and his postdoc, and that was the postdoc who pointed out that's what we should do, parallel quantum pathways, and then we make the solar cells very efficient. But to me, this question, what is really behind those oscillations, wasn't really clear from the very beginning. I kept asking questions. If you were to excite coherences between the vibrational levels, how would they look like? Would they be very different from those oscillations what you see in those samples? And the answers were not really very convincing. So we did a work, actually Niklas Christensen, who was the PhD student of mine, he had moved to Harald Kaufman when this work was done, and he developed a very simple toy model where he included vibrations into the system description. It was just an excitonic picture, and our conclusion was that also vibrational coherences would lead to very analogous type of oscillations what were observed, and this would be a much more natural explanation of the observations. At about the same time, slightly later, there was another article which were pointing out the same issues, and we were invited to give a comment to that article. So we agree very much what was written there, that this is not at all so very clear that what is oscillating in those 2D measurements is electronic coherences. They can be vibrational as well, and most likely actually are vibrational, because the big controversies of the long lifetimes would not be there. Vibrations too tend to have coherence longer than electronic subsystems. So if at around 2010 it was all about electronic coherences, then there was a Physics World website based on an article in Nature Communications, which actually made now a pretty big deal around how important the vibrations can be in energy transfer. So there was a semi-popular article in Physics World, quantized vibrations are essential to photosynthesis, say physicists. So now it was vibrations which were so very important. So what is the answer? I don't think there is a very clear answer. I believe that vibrations can lead to oscillatory features in 2D, and that's the dominating 2D oscillations what has been seen. If there are electronic coherences, how to see them? That's actually one of the questions. And one of the issues here is that if you take a pigment protein like light harvesting complex, there's lots of disorder in it. So if you take one, you take another, you take a third, all these are somewhat different. If there was any evolution, not only coherent evolution, this evolution is going to be different from sample to sample, from your system to system. And I would claim that the coherent evolution is going to be very different from one system to another. This is an example of a very nice work, single system coherent dynamics, and this is just the dye molecule. This is not as complex stuff as light harvesting complex. I'm not going to be able to explain what is seen here, but if you think of this as your projection of your block vector, if it's pointing down, it's everything ground state, pointing up, everything excited state, in between you are coherently rotating. So this is that picture of the runners on a stadium, if you wish, and leading to photon echo, etc., etc., etc. From these measurements, what they did, it's 2-pulse measurement, and they detect fluorescence. And here is the delay between the pump and probe, and the fluorescence intensities. This is what is detected. And they can fish out the evolution of this block vector as a function of this time. And you can see those evolutions here. And you can see that those evolutions are different from one molecule to another, to third, fourth. They're all very different. So if you add them up, as you do in the bulk experiment, you will lose very essential information, what is needed in order to understand what is going on in coherent dynamics. You are very welcome to interrupt me if there are questions that would be appreciated. So the same group did also work with LH2, analogous experiment, 2 pulses. Actually, they have a very broad spectrum, and the 2 pulses are generated via putting a phase shift into, let's say, the frequency-dependent phase on your pulse, and then you can construct from one pulse various shapes. 2 pulses, for example, in this case, and they can vary then the phase and time delay. It's not as nice as this. I can't think that from this kind of thing you can put together something like that, but more or less. And then by varying the phase and time delay, they again see that the fluorescence intensity goes up and down, and the conclusion was about varying pathways, etc., etc. Again, energy transfer, electronic type of explanation for the observation. Right or wrong? Well, I'm not convinced that it is electronic. but I think the future has to show. So, and in the future I hope that we will be able, maybe somebody else will do it before, I don't know, to do something like this, what Andy Merkus has developed. Now it is a 4-pulse experiment. And using those 4 pulses you can construct a 2-dimensional coherent map by detecting the fluorescence. Again, you will hear more about this method today, not going into the details. So can it be done? Together with Ivan, we had a PhD student who has done single molecule spectroscopy on light harvesting complexes. It's a different type of measurement. But still, if you do the single molecule spectroscopy, one of the questions is, does your sample survive the experiment? Because you focus quite hard to get the signal out of it. So there was an evolution of those samples. What you can see here is the time of the experiment in seconds, and here is the amount of fluorescence you get out. It's happy in the beginning, but it goes down and bleaches away. And that's what tends to happen until eventually a type of cooking recipe was developed where samples actually did survive long enough, more than 100 seconds. Some of them still bleached away, but it was a good sample what can be used for this type of pretty harsh cooking, because you burn pretty hard there. So this is the system, that's the spectrum. There was a laser and the fluorescence was detected from here, and the essence of that measurement was that you rotate the polarization of your excitation, and you also rotate the polarization of the detection. And this is, the details are here. Rafael is not here. Rafael is an expert on this topic. I think half of Ivan's group knows a lot about it, and that has been up in the seminars of the group a few times. But well, here it's a low concentration sample, so that in your screen you can separate different spots which correspond to different single LH2s to detect the fluorescence. You rotate this analyzer here. There's an additional little trick what I think is very neat, that you get the 2 perpendicular polarizations simultaneously on a CCD screen, so that you can really also check that your total fluorescence intensity didn't go down, that your samples didn't start bleaching, in order to get the information in as clean way as possible. And what is then detected is the kind of signal like that. The one oscillation is here, that the excitation has rotated, excitation polarization has rotated half a circle, right? What it takes. And then you construct a figure like this. You have the excitation polarization and the emission polarization, and you get a 2-dimensional picture like that. And now the question is, if you were able to construct that type of information, can you also make an experiment where you construct the 2-dimensional coherent picture? And Katka has done some simple estimates, and as I understand, the prospects are pretty promising. During those times, with what it took to construct this picture, we should be able to also construct the 2-dimensional picture out from such a setup what is being built currently. The 2 essential differences compared to Oregon setup. One is the wrap rate here, it's much higher, and another is that instead of let's say electronically mixing and locking signals, there is an invention what Katka has come up himself, how to do it all digitally, and that should also give us some additional detection tricks which hopefully will make it to happen here. And this is the people who directly work with it, Katka, and to help us. Okay, that's the way I see it, but I'm somewhere in the background watching things to happen. Well, I don't have more to say here. The experts are going to come in now with their presentations, but if you have any questions about that, what I told.
I have question. I remember years back, Ben gave a talk when he looked at the signal from, was it from one molecule or from an ensemble of molecules? And then my feeling was that until he gets relatively big ensemble, he could not get really phase matching. That was in 3 powers photoneco.
That was the calculation, right?
Calculations, yes.
Yes, that's exactly what I mean. If you want a phase-matched signal, then you need to average over many. But now, if you detect fluorescence, then you don't need it. That's why actually you need to do this thing here. That you actually detect fluorescence instead of photon-echo. And that's why you need a fourth pulse, what acts, well, in photon-echo, the fourth interaction is the signal-generating interaction. Here the fourth interaction is a fourth pulse.
So meaning that in echo type of experiment this is simply not possible to do it on single object.
There are, one may think probably some phase modulation technique anyway, where you do it coaxial. But I think this is the most natural way of doing it. It's like, for example, in single molecule spectroscopy, you don't work with absorption, you work with fluorescence. Even though the first experimenters I understood were in absorption. But that's the fluorescence how you should do it.
One question, like when you're trying to separate whether it is electronic coherence or vibrational coherence, from scientific point of view, probably it matters, but from if we want to use coherence in anything, does it matter?
I think it does matter because how to achieve the system which either supports vibrational or electronic coherence is, I think the strategies would be different. I think it's difficult to make a system which supports long-living electronic coherences. If the conclusion is that the vibrational coherence is important, then you can forget this part. You work further with your systems which can work with vibrational coherences. I think there is this.
Also, if you initiate any sort of electronic coherence, will that not eventually lead to vibrational coherence along the time?
Doesn't need to be. It can, but doesn't need to.
In a realistic system, would it not couple that at all?
But let's say I have my atom. There's no vibrations. I still can have electronic coherence.
But the more realistic systems.
Atom is very realistic.
Yeah, not biological, at least, so many atoms.
Yeah, okay. Well,
as soon as you go from 1 to 2, then you get vibrations, right?
Yes. Because in order to excite the vibration by light, you have to have this shifted potential surfaces, right? And that has been actually one of the arguments why those are not vibrational coherences which were observed, because it was known that in chlorophyll those shifts are very small. And if you use those known shifts and looked what would be the expected amplitudes of those oscillations, conclusion is that they are very small. But they missed this little detail what what Niklas discovered, I say to say, that if your two chlorophylls are shifted in energy in such a way that this shift is approximately about the vibrational frequency, then you get an enhancement of this coupling to the vibrational level, and you can excite a vibration even if the shift is not so big in this case. I think that was well, sooner or later somebody would have found it. That was cool. That was Niklas who found it. It was very obvious. As soon as I saw him explaining it, I thought, wow, yes, that's it.